A very good day to everyone. Welcome to the video titled, From Sexual Organ to Pregnancy. For this video, we are going to describe the anatomy and physiology of male and female reproductive systems and understand how the structures and processes can lead to a pregnancy. The learning outcomes of this course include 1. Describe the structures and functions of male and female reproductive structures. 2. Understand the process of spermatogenesis and oogenesis. 3. Explain how hormones affect the menstrual cycle. 4. Explain the process of fertilization that leads to pregnancy. Female reproductive structure. The human female reproductive structures can be divided into external and internal reproductive structures. The external reproductive structures are the parts visible on the outside of the body. The structures include clitoris and two sets of labia which cover the clitoris and the vaginal opening. The internal reproductive structures are the parts located inside the pelvic cavity. The structures consist of ovaries which produce eggs and reproductive hormones. The oviducts which receive gametes and serve as the fertilization sites. The uterus houses the embryo and serves as the site for pregnancy. External genitalia. The vagina is muscular and elastic canal leading from the vaginal opening to the uterus. It is the place for penetration of the penis and transfer of sperm during copulation. It is also a canal that provides for a newborn during birth and allows for menstrual flow during menstruation period. The vagina leads to the exterior at the vulva. The labia majora, containing fatty tissue, serve to protect the vulva. Two thin folds of skin, the labia minora, surround the opening of the vagina and the orifice of the urethra. A thin strip of tissue known as the hymen partially obscures the opening in the vagina of humans at birth, but it thins with age and can usually be worn away with normal physical activity. At the top of the labia minora is the clitoris, which is made of erectile tissue and has a rounded glans or head that is protruded and covered by the prepuce, or small hood of skin. The highly innervated clitoris is one of the most erogenous points of sexual stimulation. External genitalia. The female gonads are an important pair of ovaries located adjacent to the uterus. Ovaries are anchored in the abdominal cavity by ligaments. Each ovary contains of follicles, each consisting of a partially formed oocyte encircled by nurturing support cells. These cells sustain and safeguard the semi-developed oocyte throughout much of its complex maturation process and advancement toward potential fertilization. An oviduct, or fallopian tube, extends from the uterus toward a funnel-like opening at each ovary. It is the site for fertilization. Upon fertilization, cilia on the epithelial lining of the oviduct convey the embryo down the duct to the uterus, also known as the womb, to begin the gestation upon implantation. The uterus is a thick, muscular organ that can expand during pregnancy to accommodate a fetus. The inner lining of the uterus, the endometrium, is richly supplied with blood vessels. The neck of the uterus, called the cervix, opens into the vagina. Male reproductive structure. The human male reproductive organs are also divided into external reproductive organs and internal reproductive organs. The human male's external reproductive organs are the scrotum and penis. The internal reproductive organs consist of gonads that produce both sperm and reproductive hormones, accessory glands that produce secretion to support sperm movement, and ducts that send and propel sperms and secretions. External organs. The human penis contains the urethra as well as 3 cylinders of spongy erectile tissue. The urethra is a tube that functions as a passageway for both urines to be expelled from the body during urination and semen to be released during ejaculation. During sexual arousal, the erectile tissue fills with blood from the arteries. The resulting erection enables the penis to be inserted into the vagina. The main shaft of the penis is covered by thick skin. The head, or glans, of the penis has a much thinner outer layer and is consequently more sensitive to stimulation. The human glans is surrounded by a fold of skin called the prepuce, or foreskin. Most mammals produce sperm properly only when the testes are cooler than the rest of the body. In humans, testis temperature is maintained about 2 degrees Celsius below the core body temperature by the scrotum, a fold of the body wall. Internal organs. The male gonads, or testes, produce sperm in highly coiled tubes called seminiferous tubules. From the seminiferous tubules of a testis, the sperm pass into the coiled duct of an epididymis. Epididymis is the site where the sperms complete their maturations and become motile. The sperm are propelled from epididymis through a muscular duct, called vas deferens, during ejaculation. Each vas deferens extends behind the urinary bladder, where it joins a duct from the seminal vesicle, forming ejaculatory duct. The ejaculatory ducts open into the urethra. The urethra runs through the penis and opens to the outside at the tip of the penis, called meatus. There are 3 sets of accessory glands in male reproductive system, namely the seminal vesicles, the prostate gland, and the bulbourethral glands. They produce the secretions that combine with sperm to form semen. The bulbourethral glands are a pair of small glands along the urethra below the prostate. Before ejaculation, they secrete clear mucus that neutralizes any acidic urine remaining in the urethra. The secretions from the 2 seminal vesicles contains mucus, the sugar fructose which provides most of the sperm's energy, coagulating enzymes, and other local regulators. The prostate gland secretes its products which contains anticoagulant enzymes and citrate, which further nourish the sperms. In order for a fetus to develop, fertilization must first occur. Fertilization is the process by which the male gamete, sperm, produced in the testes, fuses with the female gamete, egg, released from the ovary, to form a zygote. The zygote then undergoes a series of cell divisions and developmental stages to become an embryo, which eventually develops into a fetus. The formation and development process of sperm is known as spermatogenesis, while the development of mature eggs is known as oogenesis. Spermatogenesis, the continuous process of sperm formation and development in adult human males. Cell division and maturation occur along the length of the seminiferous tubules, which are highly coiled structures located within the testes. This process results in the production of hundreds of millions of sperm each day. For an individual sperm cell, the entire process of development takes approximately 7 weeks to complete. Stem cells that give rise to sperm are located near the outer edge of the seminiferous tubules. As these cells progress through the spermatocyte and spermatid stages, their progeny migrate inward, ultimately leading to the release of mature sperm into the lumen of the tubule. The sperm then travel through the tubules into the epididymis, where they acquire motility. These stem cells originate from the division and differentiation of primordial germ cells in the embryonic testes. In the mature testes, they undergo meiotic division to form spermatogonia, which subsequently generates spermatocytes through further mitosis. Each primary spermatocyte undergoes meiosis to produce 4 spermatids, reducing the chromosome number from diploid, 2n equals 46 in humans, to haploid, n equals 23. Spermatids then undergo significant morphological and structural changes as they differentiate into fully functional sperms. The structure of a sperm cell is highly specialized to support its function in fertilization. In humans, as in most species, the head contains the haploid nucleus and is capped by the acrosome, a specialized vesicle that contains enzymes essential for penetrating the outer layers of the egg. Located just behind the head, numerous mitochondria generate ATP to power the movement of the flagellar tail, which propels the sperm forward. Additionally, the streamlined shape of the sperm cell minimizes resistance as it moves through the female reproductive tract, enhancing its motility and efficiency in reaching the egg. Oogenesis, the process of developing mature oocytes, eggs, is a prolonged and highly regulated process in human females. Immature eggs are formed within the ovaries during embryonic development but remain arrested in an early stage of meiosis. These oocytes do not complete their development until many years or even decades later, typically resuming maturation during the female's reproductive years in response to hormonal signals. Oogenesis, the process of egg development, begins in the female embryo with the differentiation of oogonia from primordial germ cells. Oogonia undergo meiotic divisions to produce cells that initiate meiosis but become arrested at prophase 1 before birth. These developmentally arrested cells, known as primary oocytes, are each enclosed within a small structure called a follicle. Beginning at puberty, follicle-stimulating hormone, FSH, periodically stimulates a small cohort of follicles to resume growth and development. Typically, only one follicle reaches full maturity in each menstrual cycle. Its primary oocyte completes meiosis I, resulting in a secondary oocyte and a first polar body through unequal cytokinesis. The secondary oocyte immediately enters meiosis II but halts at metaphase II, at which point it is released from the follicle during ovulation. Completion of meiosis II occurs only if a sperm cell penetrates the oocyte. Both meiotic divisions involve unequal cytokinesis, producing smaller cells called polar bodies, which eventually degenerate. The final outcome of oogenesis is a single, large, mature ovum capable of being fertilized. Following ovulation, the ruptured follicle transforms into the corpus luteum, which secretes estradiol and progesterone. Progesterone plays a crucial role in maintaining the uterine lining to support a potential pregnancy. If fertilization does not occur, the corpus luteum degenerates, and a new follicular phase begins with the next menstrual cycle. Having explored the processes of spermatogenesis and oogenesis, which form the foundation of human gamete production, we now turn our attention to the female reproductive cycles that regulate the timing and coordination of oocyte maturation and release. In particular, the ovarian cycle governs the development and ovulation of oocytes, while the menstrual cycle prepares the uterine lining for potential implantation. These cycles are controlled by hormonal feedback mechanisms and are essential for successful reproduction. In human females, the hypothalamus plays a central role in regulating reproductive function. The ovarian cycle begins with the release of gonadotropin-releasing hormone, GnRH, from the hypothalamus, which stimulates the anterior pituitary to secrete small amounts of follicle-stimulating hormone, FSH, and luteinizing hormone, LH. FSH promotes the growth of ovarian follicles, with support from LH, and these developing follicles begin to secrete estradiol. During the follicular phase, estradiol levels gradually increase as follicles grow and oocytes mature. Although several follicles begin to develop in each cycle, typically only one becomes dominant and reaches full maturity, while the others degenerate. At low concentrations, estradiol exerts negative feedback on the hypothalamus and anterior pituitary, maintaining relatively low FSH and LH levels. During the follicular phase, the rising estradiol levels stimulate the proliferative phase of the menstrual cycle. The endometrium thickens, becomes more vascularized, and prepares to support a potential embryo. As the dominant follicle continues to grow, estradiol secretion increases significantly. Once estradiol reaches a threshold concentration, it switches to positive feedback, leading to a surge in LH. This LH surge triggers the final maturation of the oocyte. Ovulation occurs approximately 24 hours after the LH surge. The mature follicle, now enlarged and fluid-filled, ruptures and releases the secondary oocyte into the oviduct. Some women may experience a brief abdominal pain called Mittelschmerz, corresponding to ovulation. Ovulation marks the transition between the proliferative and secretory phases of the uterine cycle. Following ovulation, the ruptured follicle transforms into the corpus luteum under the influence of LH. The corpus luteum secretes progesterone and estradiol, which exert negative feedback on the hypothalamus and pituitary, thereby suppressing further release of GnRH, FSH, and LH. This hormonal environment prevents the initiation of a new follicular phase while maintaining the uterine lining for possible implantation. If fertilization does not occur, the corpus luteum degenerates after about 10 to 12 days, resulting in a sharp decline in progesterone and estradiol levels. The drop in hormone levels removes the negative feedback inhibition, allowing FSH levels to rise and initiate the next cycle. As the corpus luteum degenerates, the resulting fall in ovarian hormone levels leads to vasoconstriction of endometrial arteries. The endometrial tissue breaks down and is shed from the uterus, resulting in menstruation, the menstrual flow phase. This phase typically lasts several days and marks day one of both the ovarian and uterine cycles, coinciding with the recruitment of a new group of follicles. After understanding the female reproductive cycle, it is important to note that ovulation typically occurs around day 14 of the cycle. Fertilization may take place if sperm are present in the female reproductive tract during this time, which usually results from sexual intercourse, copulation, occurring within a few days before or shortly after ovulation. Fertilization in humans is a multi-step process that begins when a sperm comes into contact with the gelatinous envelope surrounding the egg cell. The sperm releases acrosomal enzymes to dissolve a small part of this protective layer, enabling it to reach the egg's plasma membrane. Next, membrane proteins on the sperm head bind to specific receptor proteins on the egg cell. This triggers the fusion of the plasma membranes of both gametes, allowing the sperm nucleus to enter the egg cytoplasm. The middle part of the sperm, which contains mitochondria, remains outside. Once the sperm nucleus is inside, the egg activates a cortical reaction. The vitelline layer detaches and hardens into an impenetrable barrier called the zona pellucida, preventing further sperm entry. Inside the egg, the sperm nucleus and the oocyte nucleus, each containing a haploid set of chromosomes, move towards each other and fuse. This forms the diploid nucleus of the zygote, marking the beginning of a new human life. Before we conclude today's lecture, let us take a moment to reflect on the incredible coordination and complexity of the human reproductive system. We began by exploring the anatomy of the male and female reproductive organs, each uniquely structured to support the production, transport, and union of gametes. Through spermatogenesis and oogenesis, we learned how specialized cells undergo intricate developmental processes to form sperm and ova, each carrying half the genetic blueprint of a potential new life. We then turned our attention to the female reproductive cycle, a finely tuned interplay of hormones orchestrating the ovarian and uterine phases. This cycle prepares the body for pregnancy. Finally, we walked through the fertilization process, where the journey of millions of sperm culminates in the union with a single ovum, marking the beginning of a new, genetically unique human being. Thank you for your attention. Keep questioning, keep exploring, and never stop being curious about the science of life. Thank you and bye-bye.